Today I'll be talking about Dr. Bonnie Bassler, molecular biologist at Princeton University and an investigator for the Howard Hughes Medical Institute. She's best known for discovering how bacteria communicate using a process called quorum sensing, which allows them to coordinate their behavior like a multicellular organism. Dr. Bassler has received major honors including the MacArthur Genius Award and the Wolf Prize in Chemistry. Banny Bossler was born in 1962 in Chicago and grew up in California. She earned her bachelor's degree in biochemistry from UC Davis and her PhD from Johns Hopkins University. From a young age, she was curious about how life works, spending hours outdoors exploring nature. She's described herself as someone driven by curiosity and collaboration, qualities that show up not just in her science, but also in how she leads her team and teaching her students. She believes that communication is essential, both for scientists and for the bacteria she's studying, and she's becoming a strong advocate for women in STEM and for making science more inclusive and humane. After her PhD, Bassler studied glowing marine bacteria called Vibrio harvii. She noticed they only produced light when enough bacteria were present, suggesting they could somehow count each other. That observation led to her groundbreaking discovery of quorum sensing, a chemical language that bacteria use to coordinate actions like glowing, forming biofilms, or causing infection. Her lab found that one molecule called AI2 acts as a kind of universal signal used by many bacterial species, showing that even microbes can talk in a way across species boundaries. This finding completely changed how scientists view bacteria, from isolated, simple organisms to a more complex and social community. These two images show how at low cell density, when there are only a few bacteria, the signals are weak and the genes for virulence and biofilm formation are turned on so the bacteria can survive and spread. But as the population grows, the signals build up and switch the system off, changing the bacteria behavior. The figure on the left specifically also tells us how this is happening within the polypeptide.